the theme of this is you know, mechanisms of antibiotic resistance and what do you do about it. And it's fairly simple to say that you can explain most things through evolution, and if you expose bacteria to antimicrobials, you will select for the resistant ones. If you want to do something about it, reduce the selection pressure, or at least use less antimicrobials and use them judiciously. Of course, that is ridiculously oversimplistic, and I think that is half the problem with how we look at antibiotic resistance, because we talked about how do you measure antibiotic resistance, or how do you measure an outcome? Well, is it the number of species that have a resistance gene? Is it one particular resistance gene in one particular species? Is it one particular gene in one particular mobile element? Is it one mechanism or class of antibiotics to which we're resistant? We know there are multiple mechanisms for many of them. When it comes to antibiotic resistance or antimicrobial resistance, probably the thing that people care about most is the scare story that comes out of things like the O'Neill report, which says that in 20 or 30 years' time, you'll go into hospital to have a hip replacement and you'll stand a high chance of getting a multi-drug resistant bacteria, and your implant will get sort of rejected, or the operation will fail. If you want chemotherapeutics for cancer, it won't be an option, because with an impaired immune system, you'll be susceptible to a multi-drug resistant infection. And you could argue that the thing that you want to measure is the thing that's right at the top of that pyramid. So if somebody goes into hospital and they die of a multi-drug resistance, that is the problem, okay? But it won't be multi-drug resistance that kills them. It will be one particular resistance. They will die because they didn't get the antibiotic, or they did get the antibiotic to which the bacteria was resistant, and didn't get one of the antibiotics that it was susceptible to quickly enough or early enough. So where should your intervention point be? Should it be that you make sure that a bacteria never gets to that person in the first place? So you sterilize that individual before you operate on them. You operate them on in a particularly clean environment, because they wouldn't have got infected had the surgeon or the environment not provided that antibiotic gene and resistance gene. Or is it a case of making sure that if you do get an infection, that you can identify antibiotic susceptibility much, much earlier than we do now? There are multiple ways in which you can approach this problem and deal with it. And yet we talk about antibiotic resistance as if it is one particular issue. It isn't. It's like crime. And the crime that we're most worried about is murder. We don't want people to die. But actually, there are many, many things that contribute, not the least of which is human behavior. So we haven't really talked a lot about the economics and the social elements to do with it, but they are important. And there is no doubt that we need things like better surveillance, need to understand mechanisms. We clearly need better stewardship. We should not be using antibiotics or antimicrobials as growth promoters. Whoever came up with that idea was misguided, but for us in this day and age to condone or fail to condemn the use of antimicrobials as growth promoter is negligent. Agriculture, or the environment, is clearly responsible for some of the burden of human antimicrobial resistance. Colistin resistance is a good example. It came to light in China. Indeed, Timothy and his colleagues were the people to first identify it. And there is extremely strong circumstantial evidence that it directly resulted from the use of colistin as a growth promoter in pigs. Why was colistin being used in agriculture? Because, as developed as an antibiotic for human use, it was renal toxic and neurotoxic. It wipes out your kidneys and wipes out your brain. Not many clinicians are keen on using it. Why did you get worried when we generated resistance? Because you'd run out of other antibiotics. You had been careless with the use of antibiotics, and colistin had become the last antibiotic of choice for certain multi-drug resistant infections. We were responsible for creating that resistance, but had that antibiotic been in human use, probably would have generated antibiotic resistance before we did. Okay? It is not a case of pointing the finger at blame. These things happen because bugs that are exposed to antibiotic will be selected for resistance. Another example is livestock-associated MRSA. We know fairly certainly, with a fair degree of certainty, that agricultural sources of livestock-associated MRSA, particularly sequence type 398, in Europe and North America, contribute to a proportion of human MRSAs and will cause life-threatening infections in certain patients. And epidemiologically, we know this from the molecular epidemiology, it's reasonably clear. What is also apparent is that it is a problem really in countries that have low levels of MRSA to start with. Livestock-associated MRSA has been undocumented and unreported in the UK, but has existed there for quite a long time. And there seems little doubt that at the point in the sort of early 2000s when Britain had very high levels of hospital-acquired MRSA, that we didn't see it in people just because it was dwarfed by the amount of hospital-acquired MRSA. In the Netherlands, in Denmark, on the other hand, where there were very low levels of hospital-acquired MRSA, livestock-associated MRSA contributed a reasonable amount. We would be negligent if we didn't talk about the environment, because clearly one of the routes out of farms and out of agriculture into people is via the environment, because that's where we throw animal waste, spreading it on fields and into very complex microbiomes, and often at sub-inhibitory concentrations which are likely to encourage the development of antimicrobial resistance. Now, I was asked about a couple of years ago to review a paper on levels or the effect of environmental pollution. It happens to be from India, but it could have been elsewhere in the world. One of these water sources had a level of fluconazole in it that was roughly equivalent of a human dose of fluconazole per litre of water. And, you know, if I hadn't had some faith in the people who are writing it, I just wouldn't have believed it was possible. It is not that this activity is condoned or legal in this jurisdiction, but that for some reason the law isn't working. So again, we come back to human factors. Why is there a law that is not enforced, or why is there a law that is not effective? And those are things that we should seriously be asking ourselves. One of the aspects of agriculture and farming is that there's a lazy assumption that it's a bit like human medicine, but a bit different, you know, different number of legs or whatever. And I think it's very important to consider what is different about veterinary species and veterinary medicine when it comes to livestock. The bottom line is that vets have the luxury of killing or burying their failures. And indeed, if I had anything that in a farm animal, it would not be worth, the value of the animal would not justify much of an intervention. So once, if I get an uncontrollable infection, it's not an issue. We just kill the patient. Most of our patients are young. They are juveniles or young adults. You do not particularly like eating 20-year-old animal, beef from 20-year-old animals, or mutton from sheep that are 6 or 7 years old. So our animals get slaughtered at that age. We do not perform operations like hip replacements and knee replacements. If they get cancer, we don't treat them with anti-cancer drugs. So there isn't that much of the high risk where multi-drug resistant organisms cause a problem. The vast majority of our use of antibiotics are on diseases that are actually easily preventable. They are what we call endemic or production diseases that occur because the animals are being housed at very high stocking densities, often with problems with exposure to pathogens, so getting rid of slurry, ventilation are the issues. And we run a balance where there is an acceptable level of disease or acceptable mortality or morbidity that makes an enterprise still profitable, while at the same time maximising the profits that we get by putting as many together as possible. And while we live in a free market with relatively light regulation, that will always be the case. And it's very difficult for a country's government to justify very strict regulation of these things when there is a global market in feedstuffs. When our animals get diseased, we very rarely do diagnostic microbiology because it is expensive for the individual. And if we did get the result, it probably would make no difference to what we did. If it was a serious problem, we just slaughter out the entire herd and start again. And when we do diagnose disease and we do some diagnostics, it's about the group of animals, not the individual. I realize that's my 10 minutes. So, do these things contribute to disease? Yes. But I have one last thing that I think is important to it. So, I did some work in China recently. These are some preliminary results. We found MRSA in pigs, large numbers of sequence type 9 and a smaller number of sequence type 398. We found no sequence type 9 in human beings. So there was no spillover of the sequence type 9 to people. And the sequence type 398, there was some. If you look at this tree, you'll actually see that's the population of humans. That's the population in pigs that are MRSA, and there's a big population that are MSSA. And the point is that you would expect these MRSA to be contributing to human burden in China, as they do in Europe and North America, but it doesn't appear to be happening. These are preliminary results, so that, things may change. And so what do I think we should be working on? We should concentrate on the social dimension, as has been done in recent funding in India. If we improve animal health, we won't have so many ill animals, we won't be using so much antibiotic, and we need to understand AMR gene dynamics within individual species, within populations, within mobile genetic elements. It is very, very complex, not a simple answer. And finally, we need to avoid lazy assumptions, which I think is easy to fall into with such a broad topic. Thank you, and apologies for it.